the live practice session for the course Applied Environmental Microbiology. So this we are in the third week now. So if you have any doubts from previous session, you can feel free to post it in the chat box. And as far as the third week is concerned, the course content of the third week includes bioenergetics and metabolism and microbial biodiversity. So we will solve some questions based on these topics today. And before that, can you please confirm if the audio is clear as well as if you can see the screen? Am I audible? Can you please confirm if I'm audible? Aishwarya, I'm not able to hear you clearly. Are you trying to say something? Can you post in the chat box if not? Are you able to type something in the chat box or okay, I'll try to post the first question for today. You can try answering the question. At least that you can. I think the screen is visible because I don't see any issues from my side. Uh, so in that case, I'll just post the first question for today. So the first question is a microorganism switches from using NADH to FADH2 as its primary electron donor during respiration. What is the most likely consequence? Okay, so the first question is a microorganism switches from using NADH to FADH2 as its primary electron donor during respiration. What is the most likely consequence? So you can post the right option in the chat box. If you know the reason, you can also post that as well. Okay, so one person is telling that there will be no change in the ATP yield due to the proton motor force compensation. So PMF stands for proton motor force. So for this, you have to think about the complex electron transport chain complexes. So you have different complexes through which this electron transport happens, and as a result, and simultaneously, there is also a proton gradient uh, based movement, and then finally ATP synthase will be there in the series. Excuse me. So finally there will be ATP synthase in the series, and this will um synthesize ATP. So if you remember the order of that reaction, the first complex in the first complex you have the entry of NADH, and in the second complex you have the entry of FADH2. So if organism is going to switch from NADH to FADH2, it will completely skip the complex 1, and the transport will start from complex 2 only. So in this case, uh, complex 1 pumps around 4 protons, but this will not happen if you are going to use this FADH2, as it is going to bypass the complex 1. So in that case, your ATP generation is going to be less. So if you have read about glycolysis, when at the end when you are going to calculate the ATP, you will attribute some 2.5 ATP to NADH and 1.5 ATP to FADH2. This is because of this difference only. Like finally you will be calculating the number of NADH and number of FADH2 that is being formed in the glycolysis process, and then TCA cycle, then electron transport chain. Like you will subsequently study this. But at the end of glycolysis, you will make one calculation where you say that from one NADH you will get around 2.5 ATP, whereas from one FADH2 you will get only 1.5 ATP. There is So when it is going to shift to FADH2, their ATP yield is going to decrease, and the reason for ATP yield decreasing is because you are going to bypass the complex 1. In complex 1, if there is NADH, then it goes to the complex 1, but if there is FADH2, it directly goes to the complex 2. So the answer for this question is decreased ATP yield due to bypass of complex 1. I hope that is clear. So with this, we'll go to the next question. Why is complex 3 considered universal among respiring microbes? Sorry, the first option should have been it directly produces ATP, not AT. So the mentioned answer is right. So complex 3, we call it as the cytochrome BC1. So here it is also known as ubiquinol cytochrome C oxidoreductase. So in that case, the role of it is uh, to provide a link between the quinone pool and cytochrome C. So, and this connection is very important uh, because this is the main form of energy transduction in many aerobic organisms. So that is why it is considered universal among living organisms. The role is it connects the quinone pool to cytochrome C. If you see other options, it is not directly producing ATP, and the terminal electron acceptor is oxygen. So the operation obviously depends on oxygen, but this complex C only is going to couple the quinone pool to cytochrome C, and that is why it is considered universal. And this coupling is very essential in many of the organisms. So the next question is, which statement best explains why archaeal lipids differ from bacterial lipids? I mean the lipid bilayer or the cell membrane. Okay, so the primary differences, archaeal lipids have isoprenoid chains, whereas in bacteria we have fatty acid chain. And also the linking is also kind of different here. So isoprenoid chain means it is branched hydrocarbon. Uh, so it's kind of branched hydrocarbon, whereas fatty acids you cannot see that branches anyways. And also there is a difference in their bonding, which we'll see in the another question as well. So during that time I'll give more explanation for this answer. Now the next question is, what is the primary metabolic significance of acyl carrier protein? Okay, so the answer for this is, it is anchors fatty acid synthesis intermediate. So if you remember the beta oxidation pathway, this acyl carrier protein is a highly conserved small protein, and the role of this protein is to carry the fatty acids or the acyl intermediates to the extending chain of fats. So fat, is a polymeric chain. So this protein called the acyl carrier protein, what it does is it carries the acyl or the fatty acid intermediates for the formation of a long chain. So that is the primary role of acyl carrier protein, and this is involved in the beta oxidation pathway. Okay, why does the proton motor force generation resemble bacterial flagellar rotation? We also had a quick intro about this in the previous session. So both of them are driven by proton flow. So the driving force for both these is the proton motive force. So in both the cases, what happens is there is movement of protons across the electrochemical gradient, and this generates enough energy for the flagellar motion, uh, as well as ATP generation, as we saw in the ATP synthase thing. So the common thing is both are driven by the proton flow. So the next question is, which metabolic pathway contributes most directly to the biosynthetic precursor generation? If you know each of the cycle, then this question is answered. Okay, so this is again right. So the citric acid cycle, or the Krebs cycle, or the TCA cycle, tricarboxylic acid cycle, however you want to call it, it is the central pathway where you have this generation of biosynthetic precursors. Okay, so the reason is because it is kind of a connecting pathway between both catabolic and anabolic reactions. So there are generation of many intermediates that stand as precursors for other synthesis. So for example, we have this fatty acids generation, like where we have acetyl CoA and citrate, which can help in the fatty acids and cholesterol synthesis. And you can see that alpha ketoglutarate and oxaloacetate are precursors for amino acid synthesis, like for synthesis of glutamate and aspartate. So like that, you have many intermediates covered in this citric acid cycle, which act as precursors for um, other biomolecules to be generated. So if you know about the pathway, then you can answer such questions. So you remember that TCA cycle is kind of a central cycle that links your catabolic as well as anabolic reactions. Okay, so the next question is, the proton motor force consists of two components, which is, what are the two components? One is already given. proton motive force or PMF is the central thing in the electron transport chain. So here del pH refers to the um, difference in the proton concentration that drives the proton movement across the gradient. Okay, so there is difference in concentration of this proton, and that is why there is also a movement of this proton across the membrane. There is one more thing, which is the membrane potential or the potential difference. Exactly, electrical potential, membrane potential, or the potential difference. So this is the charge difference. So when you have protons on other side more, so it is also a chemical gradient as well as the electrical gradient. So with pH I have talked about the chemical gradient, so the next one is going to be a electrical gradient, and that's why this is kind of a electrochemical process. So together you have electrochemical gradient, one part is the electrical gradient and the other part is the chemical gradient. Okay, so we saw about the importance of complex 3 some time back. So complex 3 transfers electron from where to where. We saw that it is kind of universal and in aerobic respiration and stuff. So actually from where to where does the complex 3 transfer electrons? Exactly. So it's from the quinone pole to the cytochrome C, and that is what was the reason for which it was universal. Okay, so the reversibility of ATP synthase allows ATP hydrolysis to. This is a very simple question. I'll just. So this was just to introduce you to the concept of reversibility of ATP synthase. Okay, exactly. So the reversibility of ATP synthase allows ATP hydrolysis to pump proton across the membrane. it can be either ways depending on what is the need for the cell. So the next question is our kind of assertion and reason question. Archaeal membranes are more stable under extreme conditions. Ester-linked fatty acids are chemically more stable than ether-linked isoprenoid lipids. You can just tell if both the statements are right or wrong, if any of it is wrong, so that you can mention. You can just mention as A and R. You can just write like A and R are true, or A is right, R is false, something like that. Okay, so what is the mistake in R? Right. So here A is true and R is false. So here the fatty acids are linked by the ether bond, whereas the isoprenoid lipids are linked using ester bonds. So the terms are interchanged here. So the difference is that only if it is written as uh, ester-linked fatty acids are chemically more stable than, sorry, ether-linked fatty acids are more stable than ester-linked isoprenoid lipids, then the statement is right. And that is what is giving archaea the stability, and that is why you can find archaea in extreme conditions. Rather, you don't find bacteria under extreme conditions. And this was the whole point of having a three domain classification as bacteria, archaea, and eukarya, as we saw in one of the previous sessions. Okay, so the next question is kind of a match the following question, where you match the different components of the electron transport system with their role. I'll give you a couple of minutes again more to do this.
We have to give the answer also.
Okay, so we'll see the answer for this. So here, as we saw in one of the questions, like if the first question was what if the there is shift from NADH2 to FADH2, will there be any reduction in ATP and stuff? That was the first question that we saw today. So we in that question itself we saw that your complex 1 is associated with NADH. So if NADH is going to enter the system, then it enters through complex 1. If FADH2 is going to come to the system, it is going to come through the complex 2. So that is why when NADH is there, you get 2.5 ATP, whereas when there is FADH2, you get only 1.5 ATP, because the proton movement is also decreasing in that case, right? So here we see that for complex 1, we match it with 2, where it is proton pumping and NADH oxidation, whereas in complex 2 you have this FADH2 oxidation. And when you come to complex 3, we have extensively seen about complex 3, and we know that it is going to transfer it from quinone pool to cytochrome C. And complex 4 again is the tricky part. So you obviously know ATP synthase is going to do the ATP synthesis. So E to 5 is so sure. So and then you know that in complex 4 you have this reduction. Even though if you don't remember, you can go this way, but it's very easy to remember that there are 4 complexes and the ATP synthase. You can remember the lipid bilayer embedded with them. So first what happens, you have this NADH coming in, next FADH2, and then from third complex there is movement to cytochrome C, and then the fourth complex is going to form water. So that is what you call oxygen reduction. Oxygen, electron and proton together forms water, and then finally ATP synthase is going to synthesize the ATP. So this is the order. Don't get confused with this. Okay, so next is one problem for you to solve. So yes, I'll give you around 5 6 minutes to solve this. So you first find out the formula and you solve this. If you have any doubts, please post in the chat box. solve the question. So you have to use the del G formula here, which we saw in the last class. If you are facing any difficulty, please let me know in the chat box. Also, please let me know if you are trying to solve this. If you need some more time, I'll wait, or if you are not able to solve, please let me know so that I'll just go about discussing the answer. Okay. So the formula that you have to use here is del G is equal to del G naught plus RTL in Q. So that is the first, the overall formula that you have to use. So with this, you can try to solve. I'll also wait for some more time because this is kind of a mathematical problem. So if you, there is one answer already. So we'll see the answer for this question. So as I told you, the formula to solve is del G is equal to del G naught plus 2.303 RT log K, or del G naught plus RT ln Q. Okay, so that's the formula. So in this formula you now don't know what is del G naught actually. So what is given, you have the K equilibrium, you have at what temperature, at what pressure the reaction is being carried out. So the reaction is conversion of glucose 1 phosphate to glucose 6 phosphate. This you might have seen in the glycolysis pathway. Okay, and you know the concentration of glucose 6 phosphate, which is the product, and glucose 1 phosphate, which is the uh, reactant. So here there is formation of glucose 6 phosphate from glucose 1 phosphate, and you have uh, the concentration of the reactant as 3 into 10 to the power minus 5 moles, and for the product it is 10 to the power minus 4 moles. Okay, so first, in order to find del G naught, you have this formula called minus 2.303 RT log K equilibrium. Okay, so where are we getting this from? So del G naught is the value of where, when del G is equal to 0, del G naught is equal to minus 2.303 RT log K equilibrium. So this is at standard condition, that is the standard Gibbs free energy. And for standard Gibbs free energy, you have all the values here. You know what is R. So here we have everything in calories per mole. So owing to the following of that units only, you have this R value as 1.98. And the temperature, standard temperature, so you are taking 298. Okay, 25 degrees Celsius you will take. And you know this K equilibrium, so you are taking the log of it, and then uh, when you have this del G naught value, you will again go about finding the del G value. So in order to find the K of this equation, you have the concentration of the product divided by the concentration of the reactant. So with that, the value will come around 996. Okay, I guess there is a small mistake here. I've given the K equilibrium value as 19, whereas I've taken it as 17, and that is why there is difference in this calculation. Apart from that, the approach is this. The formula that you have to use here is del G is equal to del G naught plus 2.303 RT log K. And first you have to find the Gibbs free energy, standard Gibbs free energy, and from that you have to find the Gibbs free energy for the given reaction. So the next one, I hope that was clear. If there is any doubt, please post in the chat box, and when I'm uploading this PPT, I'll just change the value. So the next question is, students treat 100 unidentified environmental bacterial cells on a plate which had grown exponentially to 1 lakh bacterial cells in 2 hours. What is the growth rate constant of these bacterial cells? So again, I'll give you around 5 minutes to solve this and post in the chat box. If you are facing any difficulty in identifying the approach to solve this problem, please let me know. Okay, we'll just check the answer for this question. It is given that we have a duration of 2 hours. So you know, like, if the growth from 100 to 10 to the power 5 cells is going to take 120 minutes, number of generations you can find. And again, for this growth rate constant, can anyone tell the formula? I told that it is exponential growth. You can generally model the equation. Okay, I'll myself explain. Okay, right. Nt is equal to n naught ut. So you can, as per the thing that we have done here, you can take n is equal to 3.3 log n by n naught, and then you can find the number of generations, and mu is equal to n by t. That is one way of finding the answer. uh, you can also substitute and get it like based on the number of cells, which is log 10 to the power 5 minus log 100, which is 3, and again divided by 2 into 0.301. So that will give you 4.98. But here the calculation method that I have used gives you the answer of 4.95. So the approach is, first you are going to find the number of generations. In order to find the number of generations, the formula is 3.3 into log n by n naught. So if you are going to do that, you will get it as around 9.9 generations. Then the growth rate constant is mu is equal to n by t, which is the number of generations per the total time. So 9.9 generations in 2 hours. So mu is equal to 4.95 per hour. You can use any of the approaches that you have. So because you are using log scale, there will be difference in the answer. Okay, so that was some two problems that we had to solve for today. And next we'll again go back to uh, non-numerical questions only. So the next question is, why do purple bacteria require both ATP and purple sulfur bacteria, what we have is again purple sulfur bacteria is an anoxygenic bacteria, so it performs anoxygenic photosynthesis, but it uses only a single photosystem. So what happens is they utilize the cyclic electron flow, and this will obviously produce ATP. And as we saw, there is chemiosmotic process through which ATP is produced. But uh, unlike oxygenic photosynthesis, they in like you can see that in some plants. Okay, so that is also cyclic pathway, and produces NADH, whereas in this is anoxygenic and you cannot see any production of NADH in purple bacteria. So that is why the cyclic flow here will produce ATP and not NADH. So that is why you need both ATP and NADH for photosynthesis here. So the next question is, why is the Z scheme essential in oxygenic photosynthesis? Okay, the answer again is B here. So when we talk about this Z scheme, uh, so what happens in a Z scheme is we use the light energy so that there is movement of electrons against the thermodynamic gradient. So in photosynthesis, we need to move the electrons from water to NADP plus. Okay, this is against the thermodynamic gradient. So for this we use two things. One is the photosystem 2 and another is the photosystem 1. So in each of the photosystem, light energy is absorbed, and this will energize the electrons. And ultimately, why are we energizing the electrons? In order to reduce the NADP plus to NADPH. So this is kind of a non-cyclic flow. Z scheme, you use photosystem 1 and 2 and it is a non-cyclic flow. Here you produce ATP and NADPH, and there is need of energy in the system, and that is why we do this. So here the Z scheme is essential in oxygenic photosynthesis because the Z scheme only allows the electrons to move uphill energetically. So otherwise there is thermodynamic limitation for electrons to get energized and then reduce NADP plus to NADPH. So that is why we use the Z scheme, where we have photosystem 2 first and photosystem 1 next. Both absorb the light energy and then energize the electrons. The next question is, why is chemolithotropy particularly important in extreme environments? ensure that there is abundant organic carbon, right? So in chemolithotropy, the energy is obtained from oxidation of inorganic compounds. Let's say take some example for an extreme environment like hydrothermal vents or deep surface subsurface areas. All here organic carbon is very scarce, and in some cases it is also absent. Obviously oxygen is also mostly not present in such environments, and light is also not always present. Okay. So here what happens is there is presence of inorganic compounds only, and only from this inorganic compounds the microorganisms should derive its energy. That is why they have adapted to become chemolithotropes, where they can use this inorganic compounds as their energy source and then survive. So one interesting reason why inorganic compounds are present there is again, uh, inorganic compounds are like mostly very stable, and hence they can be present in this extreme environment. Like you compare the presence of archaea and bacteria, you compare the presence of inorganic and organic components. So archaea is stable and inorganic components are again stable. So the same analogy you can use to understand this concept. So what happens is in extreme environments you don't have much of organic carbon, oxygen or light. So the bacteria should rely on inorganic components only to derive its energy, and that is why it takes the route of chemolithotropy for survival. So the next question is, why is epiduralist oxidation sensitive to pH and oxygen levels? So the answer for this is the competition between oxidation and biological oxidation. So if we want the biological oxidation process to happen, then the chemical oxidation should not be rapid. So in that case, when and you know that metal have different ionic states in which they exist. So at neutral pH around 7, if there is more oxygen, then the chemical oxidation process is going to overtake the biological oxidation process, thereby forming insoluble precipitates of iron. And hence, since because there is formation of this insoluble precipitates, it is no more like dissolved in the system, and the biological availability or the bioavailability for the microorganisms to oxidize and take up the iron is going to be very very low. Whereas when you go for some lower pH, like at acidic pH, the chemical oxidation is not going to be very rapid. It's going to be very slow, because this Fe2 plus at that acidic pH is very stable. The oxidation is very very slow. In this case, the biological oxidation can overtake the chemical oxidation process. Uh, so that is the trade-off for any metal remediation, any bioremediation of metal. All you have to remember is the oxidation state of that metal. Okay, so what happens is, at some pH there is going to be precipitation of that metal, which means that the metal is no more bioavailable for the microorganism. I hope the answer was clear. If there are any doubts, you please post in the chat box. The next question is, why Anamox bacteria is ecologically significant? Again, this is kind of a simple question. You can just tell about the process in one line for answering this in the chat box. You can also type what is Anamox in the chat box as well. Okay, so ammonia is directly converted into nitrogen, and that process is called Anamox, because they convert ammonia into nitrate. Okay, so the answer is, Anamox process is anaerobic ammonia oxidation process, and this converts ammonium and nitrite directly to nitrogen gas. So here you are bypassing the nitrification denitrification process itself here, and that is why it is ecologically significant. Like in wastewater treatment and all, you go for when you go with this ammonia rich wastewater or something, you go for this Anamox process, and Anamox itself stands for anaerobic ammonia oxidation, and it can directly convert ammonium and nitrite directly to nitrogen. And they do not convert ammonia to nitrate, but instead directly nitrogen gas. So the next question is, which feature best distinguishes phototrophs from chemotrophs? If this question is based in clear. energy source. Okay, so we talked about different photosystems, the Z scheme and stuff. So here the question is, why is the cyclic electron flow evolutionarily advantageous? Obviously, the reason is because in the cyclic electron flow, you generate the ATP without reducing power imbalance, uh, which means, as we saw in one of the previous questions, like we are bypassing the NAD plus NADPH usage. So that is what is mean by without reducing power imbalance, right? So in that case, cyclic electron flow is evolutionary advantageous because it generates ATP but without reducing power imbalance. Okay, in this is kind of a summary question of all this that we saw. In purple bacteria, cyclic electron flow generates what but not what. We have been seeing this for the past 2 3 questions, I guess. Exactly. So in purple sulfur bacteria, you have the cyclic electron flow where ATP is produced whereas NADH is not produced. So the concept that you have to keep in mind to answer all these questions is that you have your cyclic electron flow. When you compare it with the linear electron flow, uh, there will be high energy demand. So in that case, what happens is the cyclic electron flow helps the cell meet the energy demand by synthesizing more ATP and not NADH. The next question is, in oxygenic photosynthesis, what is the electron donor? photosynthesis, your electron donor is water, and your terminal electron acceptor is going to be oxygen here. Okay, next we summarize the whole topic by having match the following. So we have different uh, processes here, and you can match them with the corresponding Okay, so the answer is for anaphysenic photosynthesis, you have no oxygen evolution in that, and oxygenic photosynthesis uses water as electron donor, as we saw in the previous question. Chemolithotropy also we have discussed before. It is present in organisms that use inorganic electron donors. And then we have nitrification, which is a two-step oxidation process, and NMOX is a direct nitrogen formation reaction. Also we saw earlier. So in case of NMOX, um, you get ammonia and nitrite directly forming nitrogen, whereas nitrification is a two-step process. Okay. So the next question is, what is the light called chlorosomes. And then nitrogenase is an enzyme and its activity is irreversibly inhibited by what? We are just talking about some compound or component that is going to irreversibly inhibit the nitrogenase activity itself. So denitrogenase is not going to inhibit nitrogenase in irreversibility. Right. So first of all, nitrogenase is a enzyme complex. Okay, and this enzyme complex is used in the reduction of nitrogen gas to ammonia, and this process we call it as nitrogen fixation process. So we talked about denitrification, nitrification, NMS, all of it. Now the other part of it is fixing the atmospheric nitrogen to ammonia. So in this process you have an enzyme called nitrogenase, right? So and this enzyme is highly sensitive to oxygen as we told. So because this enzyme is basically made of some iron sulfur clusters. Okay, so this is oxidized and inactivated. This is basically about the biochemistry of the enzyme itself. So oxygen can oxidize the iron sulfur clusters and other cofactors that are essential. So by which the enzyme itself, enzyme or the enzyme is a protein. So the protein structure and function itself gets disrupted because of oxygen. Why? Because clusters and cofactors present are oxidized. If they are oxidized, they won't perform this fixation process. So the next question is about magnetotaxis. So magnetotactic bacteria contain intracellular structures called. Again, this is a very easy question. I just wanted to introduce you to the concept of magnetotaxis, and that is why this question was kept. Exactly, magnetosomes. Prosticate bacteria increase what? So, and this is kind of an open-ended question only. So you can just write about what is a prosticate bacteria. nutrient condition, what they do is they form some cellular extensions, which obviously of the cell wall and cell membrane. So since they form the extension of cell wall and cell membrane, they try to increase the surface area to volume ratio. Thereby, they can have a better nutrient uptake. Okay. So in the last class, we saw about this diffusion and other transport mechanisms, and there we related why an increased surface area to volume ratio is required. What are the consequences of having a higher surface area and low volume to a lower surface area and high volume? So how does this affect the transport process and nutrient uptake and all of the cells? That we discussed. Based on that, this prosticate bacteria are a group of bacteria which have higher surface area to volume ratio because they form cellular extensions of cell wall and cell membrane. This allows them to efficiently uptake nutrients, and henceforth allowing them to thrive in low nutrient conditions. So that is the whole concept. So in the previous thing we have seen about nitrogen cycle, and then magnetotaxis was one concept that we saw, and now we are seeing about prosticate bacteria. So all these questions are just to make you think about and know about the different roles that microorganisms have in the environment or evolution, and kind of remediation which we'll be seeing in the future as well. So we have a couple of more questions. We'll just see them and then go. Okay, why? So the next question is, why are purple non-sulfur bacteria considered metabolically versatile? All these questions, maybe you know the answers, but it's just about thinking at that point and not getting confused with the other topics. You have to distinctly know what is the difference between purple sulfur and purple non-sulfur bacteria, how the energetics and metabolisms work in each of the bacteria and stuff. Okay, can you just give a couple of examples of what you mean by switching metabolic modes? act as chemoautotroph. So it can oxidize inorganic substances for energy and carbon dioxide for carbon. So the carbon source is CO2 and energy source is inorganic substances. So it can switch between photoheterotrophy, photoautotrophy, chemoautotrophy, and chemoheterotrophy. And as you rightly mentioned, the purple non-sulfur bacteria are tend to be metabolic chameleons. Okay, so the next question is an assertion here is the functional similarity often does not imply phylogenetic similarity. Again, we are seeing it from a evolutionary point of view. The reason is convergent evolution and horizontal gene transfer are common in microbes. When you understand what is convergent evolution and horizontal gene transfer, you can just tell if the assertion and reason is right or not. Okay, so in this question, we talk about functional similarity and phylogenetic similarity. Let's say we have two organisms, microorganisms in an environment. Okay. So we have two microorganisms. If they are functionally similar, they are going to perform similar metabolic reactions, and that is what we call it as functional similarity. Whereas phylogenetic similarity relates to how closely related both the organisms are in terms of evolution. Evolutionarily closely related organisms are said to have phylogenetic similarity, whereas the organisms that perform similar metabolic reactions or perform some tasks, they are called as functionally similar microorganisms. So in that case, functional similarity does not imply phylogenetic similarity. It is true. Just because they perform the same function, it need not mean that they are genetically very similar. So the assertion is true here. When we come to this reasoning part, we have two terms. One is the convergent evolution and another one is the horizontal gene transfer. So when we talk about convergent evolution, the term itself is self-explanatory. So two organisms are evolving and they are trying to gain or evolve having similar traits. So, when will two organisms try to evolve in a similar manner just because they are present in the same environment? Let's say we are trying to acclimatize two different microorganisms to some stressful condition. Both of them are being put into the stress, and both the organisms will try to overcome that stress and survive in that environment. So, in that case, they will try to develop similar metabolic pathways or genes responsible for similar metabolic pathways, and this phenomenon is known as convergent evolution. So they try to acquire similar functions, similar traits, uh, because they are being subjected to the similar environmental conditions. So this is known as convergent evolution. Another statement is horizontal gene transfer. So there are two types of gene transfer, vertical and horizontal. Vertical, as you know, is the gene transfer from the parent to offspring. Whereas when we talk about horizontal gene transfer, it is not from parent to offspring, not from one generation to another, but between different organisms that are present. Like there is a free gene transfer between two organisms that are present in the same environment. So this horizontal gene transfer can make one bacteria obtain a gene and henceforth a function. So the gene is going to express into some protein, and this protein can be functionally beneficial for the microorganisms which has obtained the gene. So by this phenomena also there can be transfer of the functional genes from one organism to another. It doesn't mean that both the organisms are phylogenetically similar. They have just shared the genetic material through horizontal gene transfer process. So convergent evolution and horizontal gene transfer are common phenomena in microorganisms, and they do not confer any functional similarity or phylogenetic similarity, right? So in that case, both the assertion and reason is right, and the reason explains the assertion. So again, we have a question about magnetotoxis and match the following, and the explanations are also here as follows. So with this, we'll end the session for today. If you have any doubt, you can just post in the chat box. Or else, thank you so much for joining the session. Hope to meet you next week. Thank you for joining the session. I'll just close the session now.